So we're going to start by talking about DNA structure in this video. So I wanted to make sure we had a good image of DNA. This one's a little bit cartoony, but it'll get across the different points about DNA structure that we need to. First, that DNA is made of a double helix. That means it has two sides to it. That has one side here and then one side here. Those sides are made of deoxyribose sugar and phosphate groups, like we talked about in the nucleotide in the last video. And then it is made of these nitrogen-containing bases that are forming base pairs. So if you think about DNA as a twisted ladder, then these are the rungs or the steps of the ladder, and they're produced by these nitrogen-containing bases. So there are some base pair rules that we need to make sure we follow. First, that thymine and adenine always bond together, and cytosine and guanine always bond together. The things that hold them together, what we mean by bonding, is we're talking about hydrogen bonds. And each pair has a slightly different number of hydrogen bonds. A and T always form 2 hydrogen bonds, and C and G always form 3. So in this diagram, they're representing that by these little dots here. And like I said before, we have these backbones, or the sides of DNA are made of alternating molecules of deoxyribose sugar and phosphate. So in this diagram, this dark blue color here that they've identified with a D, that's the deoxyribose sugar. And then this one here is, this lighter color here is indicating the phosphate group. So the base attaches to the sugar. So you can never have the phosphate group attached to the nitrogen-containing base. The base has to attach to the deoxyribose sugar, not the other way around. The man who figured out the base pair rules is a man called Erwin Chargaff. And Chargaff worked in 1950, and he was analyzing the amount of adenine, thymine, cytosine, and guanine in DNA. And when he broke down DNA, he found that he got the same amount of adenine as he got thymine, and guanine ended up being equal to cytosine. So he figured out that these molecules must be paired in some way so that they had equal amounts. The base pair rules would be called Chargaff's rules, but we are more going to call them the base pair rules. And it would become really important when Watson and Crick were trying to determine their model of the DNA molecule, and they had to figure out how to make equal amounts of thymine and adenine. It's one of the problems with their first molecule that Rosalind Franklin noticed. There are 2 classifications of nitrogen-containing bases. There are purines and there are pyrimidines. Purines is the shorter word but the larger molecule. That means it has 2 carbon rings. So if we think of a carbon ring as one of these shapes, okay, so this is one carbon ring and then this is the other. Pyrimidines have 2 rings, or purines have 2 rings. Pyrimidines have only 1. Pyrimidines is a longer word for the shorter molecule. How to remember these? There are lots of ways. My favorite is to the 9, so like being dressed to the 9s. And adenine and guanine are both end in 9. N-I-N-E, so they are dressed to the 9, so they're purines. And only the Y's. Pyrimidine is spelled with a Y, cytosine is spelled with a Y, and thymine is spelled with a Y. So only the Y's, dressed to the 9s. There are others like cut the pie. C and T for cut, and then pi for pyrimidines, and pure agony. So they're purines, and they have AG, so pure agony. Any of those are fine. Just make sure that you have some kind of system for remembering these. DNA likes to be equally distant. So DNA has to have the same width. So there's only enough space for 3 rings across a step of the ladder. So if we look at this picture, we can only have 3 total rings. So adenine would have 2, and then we would have thymine. So if we look at how these are linked together, we can see here's our deoxyribose sugar. That's what they're showing you with these little flags right here. And then here's thymine and adenine, and they are hydrogen bonding right here. And you can see they're the same space across as this set. There's 1, 2, 3 carbon rings, 1, 2, 3 carbon rings. This is a more detailed explanation of the nucleotide. Remember, we have a phosphate group, a deoxyribose sugar, and a nitrogen-containing base or a nitrogenous base. So, a single nucleotide is 1 sugar, 1 phosphate, 1 base. So quickly look at this image and figure out how many nucleotides we are looking at in this green box. There are 4 nucleotides in this green box. You have 1, 2, 3, and 4. And remember that DNA is made of these linked together. So here we have our nucleotide, and you can see it's bonded to the nucleotide right above it, and so on down the line. And the other side of DNA is made just the same way. However, it's running in the opposite direction. If you look at the way the sugar is oriented, you can see that the point of the house is sticking up here, right? It's pointed up, and the point of this sugar is pointed down. They're running in opposite directions, and we call that anti-parallel. So these are some more demonstrations of DNA molecules just to kind of give you an example. And then here's one that shows you the hydrogen bonds. So our next video is going to be all about DNA replication, and I'm going to leave off here so I give myself plenty of time to talk about replication.